We are again. My goodness, we're off again. The next round of Coffee Buddies. It's a marathon, and we're going to keep marathons. You keep doing, so we keep doing it. And this is going to be a fantastic one. And Coffee Buddies is a bit of a dream of us all, that it's to entertain, engage, inform, and to build a community that really shares the way of doing business. And that's our dream, and it's starting to come together. And we always surprise people by not only by looking good, but we surprise people by bringing really interesting topics to the table and fantastic speakers. And I think today is going to be an example of that. And we're moving, through the seasons. We're now becoming into t-shirt time. So, I mean, my goodness, time is moving on. And today we're going to be meandering, I would say, meandering around this idea of what's AI, artificial intelligence. How does artificial intelligence connect with the world that people like me are interested in, or have been in the wet world? And people like me, we always used to use these things called slide rules. And now we've moved into quantum computing. And today we will be hearing about something to do with that. And we've got four fantastic speakers today. And the way that we're structuring it is sort of like the big picture, going down to some super technology, coming down to some applications. And then we've got Jackie joining us. I don't know whether she's here yet, but Jackie joining us later in the session to talk about maybe the view of a drug hunter of how all of this comes together. So that's our plan. Graham's going to keep us on track as always. And I'm going to ask those questions that we all want to ask. And my great pleasure to start the event off is to introduce Patrick. Patrick is a new colleague of ours, but we were very impressed by his way of thinking. And Patrick, a pleasure to meet you again, and pleasure to introduce you to Coffee Buddies. And please, maybe you could introduce yourself, what you do, and where you do it, really.
Sure. Hi, thank you. So I'm Patrick Lilly, and I'm the CEO and founder of Liquid Biosciences. As you'll see, I'll go through a couple of slides, but we've been around about a decade in healthcare. We're a pure play analytics company based on something called mathematical evolution. And we've done about 170 major analytic projects across 44 diseases in the last decade for about 35 customers, the majority of major pharma companies in the top 10, a number of great universities, including King's College London and Oxford for our buddies Graham and Tony, and the Mayo Clinic and others. So we also, as I'm going to explain, have recently, in the last 2 years, begun partnering with a company called IV Bioholdings to spin off disease-specific companies. And I'll go through that as well.
Great. I've actually put you as co-host, Patrick, so you can share your screen.
Okay, I'll do that.
Brilliant. So are you a mathematician, Patrick?
That's an interesting question. So my education background is actually quantitative. It is economics with a minor in computer science, and then in graduate school I did a fair amount of work in quantitative finance, derivative securities, and the mathematics there. And surprisingly, that does relate quite a bit to what we're doing here.
Yeah, fantastic. Maybe you'd take us through your first slide.
Yeah, so the topic here obviously is is AI the future, true or false? There's a couple of things I would say quickly, which is, first of all, the future has to be mainstream, and we're not yet mainstream with the use of AI in healthcare, drug development, and diagnostics, but we are moving along rather quickly. And as I mentioned, for the last 10 years, we've done quite a bit of work applying our particular form of AI to healthcare with spectacular results all the way through to commercialization. So the technology that we use here, this is proprietary to us. It's in the general field of what's called evolutionary computing. And we use the construct of a countryman of yours, Alan Turing, with his ACE papers, where he mathematically proved that a series of simple math equations or logical instructions can mimic any system in nature. And so this is a fairly simple example here, but what our software does is it looks at the data and it says we have circumstances about the patient and we have outcomes for the patient. And our job is to develop an actual model, not just an approximation, but an actual model of the underlying rules that govern the system that produces those outcomes from some subset of those circumstances. And obviously knowing which circumstances, which RNAs, which genes, which proteins, which demographics and clinical history, that's very important to practitioners. This is where we start to get into AI as an augmentation for the human beings rather than a replacement. But the way it generally works is our software creates tens of millions of these Turing machine algorithms out of the raw materials of 60 or so different types of mathematical functions and relationships, as much data as you have about the patient's circumstances. You know, our sort of practical limit is about 3 billion variables per patient, but we've never seen anything more than about 28 million per patient in practice. And the last thing is constants. And it puts those algorithms into a virtual ecosystem, and they evolve, and their fitness is determined by how accurately they predict the patient outcomes. And what happens is evolution, you can think of as actually natural deselection. By treating these Turing machine algorithms as living organisms where they mate, they mutate, they migrate through separate villages or subpopulations, you wash away variables that don't matter, mathematical relationships that don't matter, and you're left with this sort of lovely residue of the things that matter, and they accurately predict the outcomes by a combination of linear and nonlinear mathematics. And that's done on about a third of the patients, and then there's a process by which we take another third to automatically select a single algorithm out of the population post-evolution. And then the final 1/3 of the patients in the data set is used for fully blinded out of sample testing. And then our process is we take that model and those factors and we apply it to another data set and others to repeatedly validate.
So just tell it, I mean, I'm interested in using patient data. Where do you get your patient data?
So it varies. So when we do work, for example, for large pharma companies, it's either their clinical trial data, or in some cases, they have access to third-party data related to disease progression, disease diagnosis, mechanistic studies for drugs and response, and so forth. So usually that is supplied by the client. Now, with our own companies that we have been spinning off with IV Bioholdings, what we're doing is we're taking publicly available gene expression and proteomic data, and we are mining those to identify the biomarkers and the relationships that predict certain outcomes in diagnosed patients. And so that is from publicly available data sets, which have typically been produced by high quality researchers under grants from the NIH, at least in the United States. And so we're able to use those to do discovery and repeat validation across multiple patient cohorts.
If I was to ask you, what particular question are you trying to answer? What question is it?
Yeah, so we've done everything from predicting treatment response, progression, drug targets, to diagnostics and a variety of things in between, like patient compliance as well. The work we're now doing in the diagnostics arena, what we do is we have a set of patients in a data set, for example, with and without a disease, or like benign versus malignant tumors in lung cancer. And our objective there is what characterizes the distinction between patients with benign tumors and malignant tumors. And that's a complex question that can't be answered by straight linear differences in biomarkers. So that is a core question for us, because at that point you can use that model to develop a diagnostic assay and put it into practice.
Yeah, so if I take a simple idea of, one population with drug, one without, and I ask the question, does the drug work? You're telling me that you can mine information that is beyond that question.
And I missed the question. I'm sorry, Tony.
As an old-fashioned, you were not old-fashioned, but as a drug hunter, I would ask the question, this population gets the drug, this doesn't get the drug, ergo the data must be showing what the drug does. You're telling me that using your methodology, you can answer different questions that I wouldn't even be able to pose. So for example, people with brown hair respond better to, I don't know, an injection of an antibody if they've got arthritis.
Right.
You can connect to the unconnected.
Yeah, one of the very first things that we did in the clinical trial space some years ago was a perfect example. There was a clinical trial where the response rate was at a particular level, and they wanted to be able to advance that for their phase 3 trial. And they also had some adverse events at a particular level they wanted to lower for the trial. And so we used baseline clinical trial data right at the time the trial started where, by the way, placebo versus treatment or standard of care versus treatment was one of the variables about the patients. And we were able to predict response, and we were able to have a separate model predicting the adverse events. And they moved that forward in their phase 3 trial to use layering on top of their selection criteria to screen out patients likely to experience adverse events and screen in more responders. So very traditional trial enrichment. And it was quite successful. It radically, decreased the adverse events and radically improved the response. And then that information was quite simple. The models could be used on label to discriminate once the product was on the market.
Okay, thank you. Shall we move on?
Sure. So I mentioned our background, that we've done over 170 projects across 44 diseases for these customers. We have been involved in a number of head-to-head comparisons, and we've always shown better results in both in terms of reproducibility and accuracy. And we've had 0 validation failures in biomarker discovery or any other arena over the last 10 years, which I think people in the industry know is quite challenging to do because there are a number of failure points. But what we've been doing for the last couple of years, in addition to serving our clients, is we partnered with a company called IV Bioholdings in the Connecticut area in the US. This was actually founded by one of our individual investors. And so we have systematized the ability to discover and validate and repeatedly validate biomarkers for diagnosis for a variety of diseases. And what we do is we create a joint venture with Marty's company, IV Bioholdings. He runs those companies, he staffs them, he funds them, and then he takes them forward into into the development of the diagnostic test. And the joint venture also has access to the causality research we do on the diagnostic biomarkers that indicates which subset of the biomarkers are more likely to be symptomatic versus closer to the beginning of the causality chain. And those that are more likely to be causal can be given to the human experts to then look at them carefully and develop them forward as potential drug targets. Or they can be provided to a company like you have on today, Benevolent AI, who specializes in that particular arena once you know the appropriate targets. The point here, and I'll show this now, is that we have systematized the discovery and validation and the sort of financing and incorporation process to such an extent that when you look at in industry, to get to the point of analytical validation right before you begin wet lab studies is typically a 6-year, $12 million R&D process. And for us, it's less than $1 million, and we do it in a number of months. It's now quite a bit less than 1 year. And we are doing several more this year, but in the past 2 years, we have done 1 per year with Marty. We did liquid lung at the very end of 2018. Last year, we did HepGene, which is in fatty liver disease and NASH diagnosis, which is a very important problem because right now it's a liver biopsy that's required, and this is a peripheral blood test. The last one that we just started, and we hired a wonderful CEO, a real rock star, sort of without the drugs and bad behavior, if you will, Liz Cormier-May, and she's running Mamagen for us, which is initially breast cancer diagnostic, especially for the high-risk groups that don't get mammograms. But it actually is a broader women's health company, so we'll be pursuing other indications in the women's health space.
Just to help me, Patrick, because I need to reframe my mind often when I see these. So year 1 to year 8 is a drug development life cycle. Yeah.
Actually, in this case, Tony, this is diagnostic development.
Okay, so diagnostics doesn't normally take 8 years, does it?
It actually does. When you look from the very beginning about the biomarker discovery and you go all the way through to commercialization, it typically does take that long. Now, what's masked is that often what's happening today is the larger diagnostic companies are acquiring smaller ones, and they are taking them at the point of CLIA validation or even analytic validation and purchasing those assets. And from there, obviously, they're only experiencing forward in the time frame, but they're not seeing all of the time that was spent up to there.
So what
part of the reason, by the way, for this time frame, Tony, is the failure rate.
But if you get 1/6 of the time, so you're cutting 5/6 out, what is it that you're improving? Which part of this process are you improving?
Well, people are suffering and dying because there isn't early detection for a number of these diseases. And in
No,
the
but
case of
no, it's not that. It's not that. What I'm getting at is what are you shaving off? Are you improving? You know, what's the shave off?
Yeah. So what's happening in sort of the traditional biomarker discovery is there's a lot of wet lab research, there's a lot of domain expertise applied, and there are a lot of failures to translate to the next step of validation. So, you know, if you look at the reproducibility rate in publications related to diagnostic biomarker discovery, more than 60% of those fail to reproduce on first validation, if they're even attempted at all. Of those that succeed in that first validation, 78% of those fail to validate on the second validation. And so there's a lot of time spent discovering biomarkers, discarding biomarkers, doing it again, and so forth. The other thing is that the process of evaluating these with the mathematics that have been used traditionally are very much linear, and they're very much holding all of these markers as sort of separate from each other as independent variables. And biology, as you know, doesn't work that way. There's a lot of complex interactions. So if you're unable to discover and model the actual biology from very, very large sets of data about the patients, you won't get to either an algorithm or the right biomarkers, and you will go through this repetitive failure process. The other thing, of course, is that our analytic method is incredibly fast. It's based on evolution in the real world, which people think about operating over millions of years. But as we think of COVID and all the mutations that have come out in response to our responses to the virus, you realize evolution operates on a very, very rapid scale. We're simply accelerating that in this sort of virtual computerized ecosystem. And so these biomarker discovery projects can happen in a matter of weeks rather than a matter of years.
Okay, interesting. Let's move on to the next slide.
So the last part, I mean, this is what I referred to, is that it's very important to us keeping in mind the patients at all times. And the reason we wanted to systematize this entire process is we perceived time, cost, and risk to be incredibly high. And, you know, every week that goes by without a decent Alzheimer's diagnosis is tens of thousands of people who may not get a treatment in time. It's the same for cancers. It's the same for endometriosis. It's the same for a variety of diseases. The other thing is using these markers to predict progression is very important, because the earlier you intervene, the better it is for the patient and the better it is for healthcare costs generally.
Excellent. Is that your end of your slides?
It is, thank you.
Yeah, maybe you could take your slides off and we can go back into the room. I think, Graham, what I'd like to do now is just invite a couple of questions from the audience. I think what I'd like, 2 or 3 questions from the audience. I love what you're saying. I mean, I think for me, it's always what context. You know, I'm a man who's an injector or a cutter, really. You know, I've done a lot of injections and I've cut a lot of things. So, I mean, I'm trying to understand where it fits in with injections and cutters, but I'm going to ask others to ask probing questions if they can. Just let me bring the group back.
Yeah,
have we got
group back? If you put your hand up, if you got a question for Patrick.
That would be quicker. Anyone got a question?
Tom has. Good man, Tom.
Yeah, you can hear me. Yeah.
Yes, we can.
We
can always hear you, Tom. Actually, that's funny. That
I know it is, isn't it? Yeah. you know the marvels of switching your mic on.
Yeah. As long as you switch it off during the break. Sorry, Tom. Carry on.
Yeah. You know, Patrick, absolutely fascinating talk. Thank you very much indeed. I've been sort of wary of AI. I quite like acquired intelligence, and AI, you know, to me is another tool in our toolbox in trying to understand, you know, the sort of, say, pathophysiology of a disease and how that, you know, relates to biomarkers. But one thing AI has been wrestling with, and you hit on this, is you're having problems with the replication crisis. Now, you mentioned that your algorithms are more sophisticated than anybody else's, you know, which is fine. But when you've got prestigious groups like MIT publishing an article in, you know, their technology review in 2020, and one coming out of Nature as well, talking about the transparency and reproducibility of data. You know, it is a problem for, you know, the platform technology. And you mentioned, you know, failure rates of biomarkers being 60% and 70% on the second time. But when you think, that is the problem with any biological research, you know, at the lab bench, you know, people trying to replicate other people's data. It is a huge problem. Now, I'm, you know, for what you're doing, but how are you tackling this enormous problem?
Well, it's incredibly thorny, as you've said. Not only there are a lot of failure points, but even within analytics, there's a tremendous number of potential failure points.
Right.
And I think I'd like to intertwine two things that you said. You're talking about reproducibility, but you also talked about transparency, right? Can we understand what the model is doing? Because to say something that is repetitively reproducible is wonderful, because then you know that it works. On the other hand, you're never going to get adoption if people don't understand why. Right. And what we care about is adoption, because without adoption, you don't help the patients. So when I talked about the framework of circumstances and outcomes, that's a very important thing, because when we think about, you know, Bertrand Russell said, a theory is nothing more than a mathematical description of your observations. So our job, taking the patient circumstances and outcomes, is not to produce a model that is just a classifier or a predictor. It actually must get to a model of the underlying sort of what we call the physics of the problem. And in fact, if you give our software a physics problem, you know, Galileo dropping cannonballs off the tower from various heights, where the input is the height and the outcome is how long did it take to fall, it will, without knowing what the problem is, come back and give you the law for acceleration under gravity, which is a very simple law, but it's also nonlinear, right? When you give that problem blinded to neural networks and support vector machines and logistic regression and so forth, they all fail out of sample. They try to approximate what observations you had, but they can't produce a model that actually works beyond those constraints. And so we approach biology in very much the same way. The other thing I'll say is we pay a lot of attention to the right data sets and getting patient heterogeneity and dividing up the patient samples so that you're not overfitting during the evolutionary process as much as you otherwise would, because this is an extraordinarily flexible method. The other thing, though, is this sort of selection and then fully out of sample validation we use gives you a very strong indication of how well it will hold up on new patients. And there's a lot in terms of how you select the algorithms out of the population post-evolution. The last thing I'll say is what ends up happening during that process is it actually causes the system to produce the simplest algorithms possible, linear or nonlinear, the simplest algorithms possible to then predict, you know, future events and so forth. And what I'll say is, for example, in our diagnostic test for lung cancer, the actual algorithm is 9 simple mathematical instructions. 3 of them are nonlinear, 1 is discontinuous, and the rest are straightforward mathematics that we're used to from grade school. So it's sophisticated in the sense that it's able to model any kind of relationship, linear and nonlinear, but it's simple in the sense that it's understandable. And once you have that model, you can literally put it into Excel or Python or any system and simulate against it and say, what happens if I, for example, lower this biomarker? What effect does it have on the patient's diagnostic outcome?
I think we've got to, I want to take one question, Graham, and then I'm going to move on. But I mean, I think a lot of us, it's language. I mean, to be honest, I have real problems with this because, you know, I come from a world of injecting and cutting, as I say, and I'm trying to understand if that thing you know, fluid I've got in my needle, you know, how does it work? And, you know, to think I can understand from maths is hard for me. But I'm going to take one more question.
Okay, we've got 2 quick, we've got more than that. But
I want to take one question. And then
okay, we'll go for Jonathan Betts just quickly.
Yeah. Where's Jonathan? Jonathan, where are you?
Hey, I'm here.
Oh, there you are. You're
Patrick, thanks so much. I mean, it's quite a simple question because I think when you present the model, it looks like, you know, that chart where you go down to 1/6 of the time and 1/12 the cost, or it may have been the other way around. It seems like, you know, the world is your oyster. You could be powering ahead with hundreds of these things potentially. So what is the limitation on that side? Is it is it ultimately down to the data sets, the patient data sets that you have? Or yeah.
Well, there's a tremendous wealth of data sets. I mean, even the NIH repository that I mentioned for gene expression and proteomics has, you know, tens of thousands of data sets we can use across all indications. So that's not really the constraint. The constraint has been more about systematizing the business and commercialization side and making sure we had reliable sources of capital, reliable sources of staff, and so forth, and then simply expanding our computational capacity. And so, you know, we've built this sort of credibility and pipeline by serving major customers. Now we're turning the wheel faster and faster on these companies. And again, the scalability is simply a factor of, can we get these to commercialization after the discovery as quickly as possible.
What I want to do is, Patrick, I want to bring in Shahar now. Shahar, are you there? Because I want to get a transversal opinion on this, you know, from another angle. So we've moved from one angle to another angle. So Shahar, maybe you could introduce yourself and tell everyone where you sit in this world.
Sure. So I am CEO and co-founder of Polaris Quantum Biotech. And what we do is we are using quantum computing, some AI machine learning, not too much of that, and good old computational chemistry to design a novel molecular drug. And I want to say two things here. First, I relate so much to what Patrick was saying, that in many cases, the problem is not finding the solution, but it is in scaling it up and making sure that you have a business case there for that solution, or not just to find the answer, but how you commercialize the answer and make sure it's working on different cases or different biologies, different indications. That's the first thing that I wanted to say
can
here.
You just give an example, Shahar, so we can ground ourselves in an example.
Sure. So what we do is we look to find a molecular drug in very large chemical space. So we start from a protein target, some kind of a disease or disease profile, and then we build virtual chemical space, huge ones, 30 billion molecules, and we find the right molecule in that. And think about this in, you know, if you're interested in a protein-protein interaction. It's difficult to find those kind of small molecules that disrupt protein-protein interactions. In order to do that, you need to search very large chemical spaces. And we do that very efficiently, very fast. But now the question comes, and this is what Patrick was saying, is that you have a solution, now find the right problem that will make your solution commercially reasonable. Okay, because I can find many kinase solutions or molecules that will block kinase inhibitor. Nobody's interested in that. There are enough of those in the market. Another kinase, you know, and you can say this is for JAK3, JAK2. Nobody cares about this. If you come and you say, I have a way to block protein-protein interaction, people are much more interested in that. You use the same technology. You got it in the same time frame. So it takes us 3 months to do that, to go from identifying a protein or from a protein target to a set of small molecules that are good, ready for binding, for synthesis and binding. And we've shown that this works consistently. The question is, how do you commercialize them? And how do you do it?
Just one question. Does Big Pharma allow you to use all their structures? Because I mean, the best library of information must be in all the big pharmas if you put them all together.
Yes, they are out. The structures are out there from Big Pharma, but I think for us, at least right now, because what we're doing is we are calculating the structure, the molecules. We do not need that data from Big Pharma. We can show that what we're doing is working now on problem where the structures are known. If we can work on known problem and solve a problem that is known that people have struggled with in the past and couldn't answer that, then the next step is going to be that Big Pharma comes to us and say, now solve our proprietary problems. Also, because we are not doing machine learning on binding affinity, we don't need those data sets, right? Those data sets, we can now have a whole discussion about how good they are and how big they are and if they are good enough for predicting binding affinity. That's a whole different discussion. But what we're doing is we are producing our own data. So we don't need to go out there and get those sets from Big Pharma.
So for you, it's almost, you've got a technology looking for a reason to use it.
Yes, I am looking for a reason to use it. I found several reasons. I think what the question for me is not just proving that my technology is the right technology to use, but also making sure that I have the right customers, and that the next step is going to be that the customers will come to us and say, now use your technology to solve our problems. So I can go and find several cases and publish those cases, but just that's going to take me a lot of time and a lot of money. What I am doing now is identifying where there's a pain point in our industry and solving those pain points and trying to make the process move faster than doing it.
What I want to do now, I want to get another view, and then I want to start getting the, if you like, the group of people talking together. Pek, I'm going to ask you now to come in. Just Pek, Pek's got an interesting background and is a lovely person, and I'd like to maybe hear from Pek, just so that we can start getting again another view of this, because in a way we're trying to create a bouncing around of ideas. Pek, just maybe from your point of view, listening to Shahar, listening to Patrick, you know, what do you do, and where contextually does it sit in your head?
Right. No, this is super interesting, Patrick, Shahar as well. I'm Pek. I'm a biologist, actually a genomics person by training, and have always thought about biology from the context of data, given my genomics background. So I'm the CEO and co-founder of a company called Oranza, and Oranza is I call it an AI drug discovery company, because we're really a technology platform like Liquid Biosciences and Shahar's company as well. And when I co-founded Oranza with my co-founder, one of the biggest problems I've always been wanting to solve is how do we get to the right drug for the right patients, and starting from the biology, because I worked for a big pharmaceutical company for a long time when, after they bought, the startup that I worked for many years ago. And I've often seen, drugs getting into the clinic, and, even though the drug is perfectly fine, it binds to the target, really well, you just can't get to the right patients, or enough right patients that you get the efficacy, that you need, the efficacy rate that you need. So Oranza was built upon that premise, is that why don't we start to understand biology first? And what is the problem? So it's very much like what Patrick was saying, is that, okay, it is all about maybe biomarkers to go find the right patients. But then the idea behind Oranza is that, well, you can find the right patients, but you also have to find the right compound. So why not marry both and make the discovery a more systematic and principled way? So we created an engine that really has multiple solutions to what I see as a problem. And the engine, first of all, actually attacks the issue of disease heterogeneity. So why do we fail in creating a drug for, say, diabetes? Well, because, let's say a certain kind of cancer. That's because there are many kinds of subtypes within the cancer. So how do we find the subtypes first and then figure out what drug or discover a molecule that's good for that? So Oranza really takes, like Patrick, we really appreciate public data, because from public data, there comes a heterogeneity that people like us appreciate, because if you were to create the most pristine data set in your lab, You go out to the wild world of clinical trials, you fail because it's not representative. So for us, we have a lot of perils, I think, with Liquid Biosciences, is that we go out and we look at public data and we say these data come from everywhere. So if we could systematically look at each of these data sets or cohorts of disease and able to find subtypes from each one of them, and then find a recurring signal for each of the subtype, then that signal, the chances of it being real, biologically relevant is extremely high, because if it's an artifact, you probably would not find it recurring in more than one data set. So the idea is behind Aranza's engine is this, but once we find that, because we want to discover drugs against those subtypes, we have another part of the engine where we have created a really big digital library of compounds, all kinds of compounds that are out there in the world that has actually met a living cell and has genomics, gene expression done on it. And the idea is then to match these compounds as a starting point for analoging, perhaps, or for discovering a biology, match these compounds to those disease subtypes in a way that it's from the biology point of view. So the idea is not, it's not new in a sense that, people in the genomics field or in the drug discovery field have often thought that if you have a small molecule or any kind of perturbation, even a knockdown, in order for you to at least be relevant to the disease biology, your gene expression better go the other way of the disease biology. So the idea behind this part of the engine is to do it that way.
So Pek, I'm going to wind in Patrick again and Shahar, so I get the three-way conversation going. I mean, Patrick, bringing your thoughts on where we're meandering at the moment.
And I was just going to add to that very quickly, but the Jackie's here as well. If you haven't seen that,
I can see Jackie. We'll bring Jackie in as well. Carry on, Patrick. And
yeah. So first of all, pretty much everything Pek said resonates with us, right? Heterogeneity is critical. Repetition of finding the particular signals is critical. Also, the approach of once you can find the biomarkers that distinguish a disease, you can sort of run that backwards and say, well, we need compounds that will put that back to a homeostatic level within the patient. And I appreciate that particular focus. And what I would say is, I intend to call both Shahar and Pek afterward, because, we are very interested in the drug development, because we're very good at identifying the drug targets and the general pathways involved in a disease from a causal basis, right? So yeah, it makes all the sense in the world what you said.
Right, I think it makes total sense in what you and Shahar said as well. And and also, if we're going to talk about how we make drug discovery, we are small companies, young companies trying to break into the pharma field, really using computation. It's not an easy process in a sense, so we decided, at Aranza, that we need to create our own pipeline, as proof of concept, as well as, being a business model. And so we actually have a pipeline that is quite healthy. We've actually already out-licensed a drug product to another pharma company, about 1.5 years ago. We're working on a liver cancer drug that is almost ready to go to phase 1. I think it is important for companies like ours who's using computation and AI. There's a fair amount of skepticism out there that maybe for the lack of funding, like you said, lack of other things, might stop at the in silico part. But I think it's important for us to carry it on.
I'm going to wind in, and we're very lucky to have Jackie with us. Jackie, you're down the bottom of my screen, so welcome to Jackie. Can you hear us, Jackie? Yeah. Can you take yourself off mute, or Graham maybe take yourself off?
Yes. Yeah. So no, I can hear you, Tony. I can hear you.
So welcome, Jackie. I know you joined late, so I was waiting till you caught up with some of the vibrations.
Yeah. No, thanks.
What's going on? But I mean, Jackie, I've known Jackie for many years, and Jackie, I think, probably has a similar brain to me, in that we've seen many drugs come and go, and we've often said at the end, well, the drug just doesn't work. And I think now, in a world where maybe we would never say the drug doesn't work, we just hunt out the population that it may work in, which could get smaller and smaller. But I mean, what I'm trying to do now, Jackie, is bring in, if you like, your optic on this. And after listening to Patrick and Peck and Shahar, what's your initial starting point with your thoughts?
Well, I mean, I think obviously, apologies, Patrick. I missed what you were saying earlier, but heard the other two. I think that we are in a situation where, you know, all of these companies are, and ourselves, are collaborating with large pharma, and I think large pharma are beginning to embrace AI at different rates, different degrees of adoption. So you've got companies like Novartis, Roche, AstraZeneca, who I think are very far along the journey. And I think there are other companies that are coming along a little behind. But I think the skepticism that existed before, I think is being dispelled, because you're seeing success stories, right? You're seeing, you know, whether it's in biomarker development, whether it's in new compounds, you know, like the repositioning, like the work that Benevolent did with baricitinib. The value of this technology is beginning to be realized. And frankly, if we look at the economics of the returns on investment at the moment of large pharma, there's two ways we can help. One is by cutting the cost of failure, because the kind of couple of billion it takes to get a drug to the market incorporates the cost of everything that failed. And the other is by improving the chance of success. And I think what we've already seen is that we have cut the cost of failure, and now we're beginning to see more examples of where we've increased the chance of success.
And is that a general, I mean, can you give us some, you know, are there public domain examples of success, Jackie?
Well, actually, I mean, in terms of target validation and identification, I think there are a number of examples. Our own is recently we announced that we've been working with AstraZeneca and have come up with a novel target that's been taken into their portfolio for chronic kidney disease. Now, that sort of target validation will take time before it realizes the data in terms of proving clinical efficacy. But the example I'd use from our own work was, as I mentioned, with baricitinib in the treatment of COVID. I mean, we identified that as a potential antiviral as well as anti-inflammatory in February last year, published it, talked to Lilly. They hadn't appreciated the potential for antiviral activity, did the assays, it went into the comparative add-on trial with remdesivir in the US and actually showed benefit.
So I completely get it for reprofiling or repurposing, but I mean, Shahar's position is, what was it, Shahar, that for two novel proteins, you can use this sort of stuff for two novel proteins? Maybe Shahar, if Jackie didn't hear, you could just say that again.
No, I did. I heard what she said, and
you heard what she said.
Yeah, from the quantum computing to use to predict the structures. And if you look at you know, the work that Schrödinger have done, I mean, one of the things that's quite interesting, because I heard you say about, you know, the pharma companies have huge libraries. When SmithKline and Beecham merged, and when SmithKline, Beecham, and GSK merged, actually there was an awful lot of overlap in the chemistry, in the compound collections, primarily because until relatively recently, medicinal chemists sort of use about 32 to 36 synthetic routes. And there's a huge scope in terms of chemical space. And one of the things that Schrödinger said is that, you know, when they've been using their platform to create new molecules, and they've done direct comparisons between medicinal chemistry and you know, the traditional medicinal chemistry approach and the computational generative approach, the computational approach always produces the compounds that the medicinal chemist would make, but also a whole range of others in new chemical space. And then the other thing that's allowed that to happen, which we're using in terms of our chemistry, is being able to use ML to really predict new synthetic routes and be able to incorporate synthetic tractability into the assessment for new molecules. So I think we're going to see a very interesting time in terms of molecular design and how that's going to translate into new molecules.
Yeah, I want to bring in Peck, I think, for just some reaction to what Jackie said.
No, I think I agree with Jackie as well, is that, I think big pharmas, are starting, have really been always dipping the, starting and really being interested. I wouldn't say dipping the toes, but yes, dipping the toes into, the area of AI and drug discovery and discovery biomarkers. I think when the genome was sequenced over 22 years ago, I think there was a, really kind of a big step towards kind of thinking about data and how to apply that into drug discovery. And I was working for Rosetta at that time, and, Merck bought us. And, there's still, hurdles around, really kind of using it to its fullest. But I think, fast forward to today, like Jackie was saying, that there are a lot more collaborations with big pharma. I think you heard Patrick say this, Nashaha, and Aranza as well. We have collaborations with big pharma, and that is really kind of, getting to the next step. I think it is, I think these are good steps towards, really kind of integrating this as it should, as an ecosystem to really have, AI technology, computers at the forefront. And it might be maybe not a kind of a hockey stick, but maybe it might be like a gradual kind of, march towards that. But I think it's happening. That's what I feel.
Yeah, I mean, I'd echo what you were saying, Peck, about the kind of, a genomic revolution with the advent of the publish of the human genome. It made people think differently about how they looked at targets, identified targets, etc. And it certainly, at the time at SmithKline Beecham, it changed, and also in the legacy Glaxo, it changed how new programs were set up. It was a different way of thinking, and I think that's going to happen with AI. And I think in many ways it's so complementary, because you've got the disease expertise within the pharma companies and the high throughput assay systems, and we can bring the AI, the kind of platform technology that can help in terms of the early part of identifying the process. But then you've got the disease expertise and the validated assays that can expedite the validation. So, as I say, in the way we work with AstraZeneca, it's worked really well. It's not a kind of either or, it's a kind of, let's bring the best of the two together and come out with something that's synergistic.
Patrick,
And I wanted to add.
sorry, Shaha.
Yeah.
Shaha, yeah.
Yes. So, but I think we are looking here at two very different things, and I'm looking at this from the data science perspective. When we think about genomics and patient populations, we have much more data than when we look at binding affinity or designing molecules. The kind of data sets that you have for genomics are in the 10,000s to billions. The kind of data sets you have with small molecules is 1,000. So I think we should differentiate these two things. And I completely understand using AI, machine learning, and data science to analyze patient populations. I am not sure I understand where does it work or how is it working so well when you're thinking about designing new molecules. And this is just the set of data that you have.
So I think this is
different.
where Peck, you've got a pipeline,
Right.
so you must design stuff. How did you do it?
Yeah, so this is a great, so we think about that as well, because we bring molecules, so we are working on small molecules, But our starting point is not a target. Usually we start from the biology. So we basically use gene expression because the idea that gene expression gives you a way to look into what the cell is doing. It's RNA activity, I talk about bias as well, but I'm an RNA person. However, I think when the DNA sequencing came about, everyone was on the DNA bandwagon, but realized that DNA, while it's good, is pretty static. However, RNA tells you about the disease and the biology. So we use that as a hook to basically draw out the small molecules. And the only way we could, that we start with, right, is to figure out what small molecules do in living cells in terms of their perturbations and in terms of from their gene expression point of view. So we are matching basically activity of the compounds and activity of the disease and seeing how they come together. And that's how Aranza finds its small molecule from a digital library, and then we analog it. However, we also have computational chemists in our team because we appreciate that finding that is only the first step. So if it could be repurposed for something, fine. But then repurposing has its own problems in the sense that maybe there is no commercial value, or maybe there is hard to get method of use IP, for example. But let's say, for example, once you find a starting molecule, you might want to make it better. And so that's when chemistry comes in. And we've actually been thinking hard about how to actually use, machine learning, AI to actually make, our medicinal chemist life easier in a sense that how do we actually then make it? So actually, to be honest, I was caught by that sentence that you talked about, Shahar, that you said, sure, we can find another kinase compound, we can find another, TKI, whatever that is. But how do you find things that are protein disruptors or protein, other protein disruptors? And I kind of thought about my own molecule, which is AU409, that we want to bring it to the clinic. We found it through biology that AU409, which is a new analog that we made from the original HIT compound, is actually a protein or protein DNA disruptor. So it modulates RNA transcription. We didn't ask for it at first. We just say, let's take a look at the biology of liver cancer and see what the machine actually gives us. And that's how we got to it. But once we got to it, we say, okay, now we know what it does. How can we make better molecules? So I think your way of coming in from a certain aspect of molecule is actually quite interesting. We've always been thinking about how to make that better. It's just that from Aranza's point of view, we always come from the biology angle. So we are trying to marry chemistry and biology together. I feel like maybe chemistry is very important, but is also limited in some ways that you start from chemistry, you don't actually understand the biology as well as it should. So we start from the understanding the biology first, and then doing the chemistry later.
Well, what I'm going to do now, I'm going to bring in, because I think we've got a great goldfish bowl.
Was there a quick something from Jackie just on the back of that? I was just wondering with her hand up.
Going to say that I think the important thing here is actually you don't look at data sets from genomics on patients separately from sort of basic biology and from chemistry. Actually, the power is incorporating all of that information and then using it together. That's that's everything.
Now I'm going to bring
exactly.
I want to bring in other views because let's bring in the others now. So I'm going to open up. We've got 10 questions in the chat line. Have we, Graham?
Well, we're going to go for Lucy. Lucy, would you like to go first?
I mean, you've heard the debate. Where's it going with you in your mind?
What's that?
Lucy, we want Lucy's question.
Where is Lucy?
So I'm a layman. I'm not really in this area, but from a patient point of view, how far are we from having our genome read to being able to find out what drugs actually will work for us? And so I'm kind of like from the industry, from a business point of view here, thinking, okay, what are we going to see in the next 5-10 years? But where are we really? And are we kidding ourselves? Or it sounds great that big pharma's actually working together now and everything, but where are we really? And are the robots taking over?
Which is always the concern, isn't it, Patrick?
Yeah, I have a comment on that. There's two things. One is, I think as an industry, we need to be more careful about emphasizing the genome to people, because as Peck said, it's static. It's the blueprint for the Formula One car. It's not the telemetry of how it's doing on the track right now. And so gene expression and proteomics are very, very important. I also agree with Jackie that the genome needs to be considered in concert with those things. And I think, you know, for lay people in particular, it's very important. And Tony referenced this with respect to, you know, cutting and injecting. You know, we had an example where, let's just forget about the omics for a second. We did work on predicting surgical complications, and set aside all the complex algorithms and nonlinearity and all the background factors. One of the things that came out was very, very simple in practice. If the patient is of a particular ethnicity and they have labored breathing in the recovery room post-surgery for this particular type of implant, they are highly likely to have an infection within a 30-day period. What that means is you don't wait for the 30-day checkup, you just take action now. And if you notice, there's an algorithm there in sort of an English language sense, and if the patient is this and this, but there's not mathematics. So I think that's important. The second thing I would say is, I want to echo what Jackie said and Peck said, is the job of AI here is, let's not call it artificial intelligence for a moment, let's call it artificial imagination. Because as experts, what we tend to do is go where we know. There's this thing called streetlight bias, where you look for your keys underneath the street lamp because that's where the light is, right? The augmentation of AI should be around discovering new spaces of the biology and the drug compounds, and then taking those in sort of plain English and giving them to the experts to narrow the number of things their brains have to consider in moving it forward. That's where the great efficiencies and discoveries will come from.
Agreed.
Right.
Okay,
I just want to comment on the question as well, is that, you know, I think we are not, I don't think we're anywhere close to, you know, reading somebody's genome and said, hey, this is the drug you should be taking. I think, you know, the steps towards that, it may be coming one day, but I think we are more, at least for, you know, for us here, I think what Patrick was saying is that using machines and, you know, and data and algorithms are really just to help us, you know, drug discoverers, you know, biologists, you know, to make drugs, better drugs for patients is for us, is like an artificial intelligence, it's augmentation of the human intelligence. That's how I see it. And you know, we are getting towards the first step of solving problems, because if you look at data points, if you look at NCBI, for example, that was created to, you know, accept these public data sets, and you look at the sequencing runs that come out from RNA sequencing or even DNA sequencing, the data is huge. You know, as a human, you can never ever be able to even look at one thing at a time, let alone 1,000 things at a time. So that's where computers and algorithms come in to help us.
What I'm going to ask my friend Tom, you know, do you think some of those antidepressants that you've been working on for umpteen years would have benefited from all of this?
Yeah, absolutely. You know, I think, as I said to Patrick earlier, you know, to me, AI, you know, is a tool in a toolbox. And unfortunately, you know, we've got some very sharp tools, and I like to think AI is one of them, and some very blunt tools. And, you know, what we're trying to do, you know, Jackie mentioned, you know, quite rightly, you know, that, you know, the chemistry in the Beecham, SmithKline Beecham, and, you know, Glaxo, a lot of the chemists, you know, we're doing the same things, working on antidepressants, very similar structures, few modifications. And, you know, from that, you know, we were basically looking, and still are after 50 odd years, you know, we're still looking for differences. And I think that's where AI comes in. So when you look at the genomics revolution in the 90s, you think of the genomics revolution, it wasn't a revolution, it was an evolution. And, you know, what we're going through now is an evolution of artificial intelligence, machine learning, to see how that combines, as Jackie said, with regard to, you know, the genome. And from you know, using 2 sharp tools, we may well come out with a product at the end, you know, which we can commercialize and treat depression, for example.
I mean, yeah, good point. I'm looking for around in the group.
Yeah, I would like to pick on Akok, please, because I know you had a question earlier, didn't you? And I know you're new to the group, so if you'd like to just introduce yourself and yeah, and then ask your question. Thank you.
Okay, thank you very much, Graham. My last name is Akok, and that's how it is written. I can see that. That's why you called me Akok.
Yes.
Perfectly
yes.
okay. It was understandable. My name is Mehmet Naji, but that's a mouthful. So you can call me Mehmet or Naji. I have a background in, I started life with 2 majors, one in electronics and cybernetics, plus molecular biology. I couldn't choose. It was science fiction at that time, both of them extremely interesting. And then I decided I'd learn some data science. So I have a master in applied statistics and data science, another master in computer science, and a PhD in computer science. And I have 2 positions, adjunct positions at 2 universities in Norway. I'm based in Oslo, Norway.
Welcome.
So I'm sort of mastered in between computational sciences. I've been teaching AI and that kind of stuff for quite some time, and data sciences and databases. And I've been working, we have a startup called InVirtualis, which is working, the name says quite a lot, I suppose. It is trying to get everything that you can do in vitro over to be doable in the virtual, in the digital world, basically. So that's why it's called InVirtualis. Yeah, that was maybe a bit about me. I think all the discussion was extremely interesting. One of the questions that I had, the original, and now I have 15 more questions because...
We'll keep it to 1 or 2 maybe, depending how snappy.
Well, I'll start with Patrick if that's possible.
Yeah.
So when I had my first question, it looked as if you had equations, Patrick, on your first slide that were sort of selected out, filtered out. Some of them were filtered out, some of them were filtered in, and some of them were slightly modified as they were filtered in. And it looked as if you had domain information in those equations. So it's, for me, as someone who's been working with AI from when AI became popular and then unpopular, almost 40 years back, it looks like you're using genetic algorithms, but also domain knowledge. How does that domain knowledge come about? Well, how do you find those attributes?
Right, right. So what I will say is we are strictly using the data with no assumptions. We're not using any domain knowledge at all, okay, except to the extent that somebody chose what information to collect about the patients.
Yeah.
Right. But occasionally we get these unbelievable data sets. For example, we've been working with Dr. Simon Lovestone for a number of years around neurodegenerative diseases. You know, Simon is at Oxford today. And, you know, we're working on a data set with him that has the full genome, all the SNPs. It has gene expression, it has proteomics, it has metabolomics and 2 different kinds of mass spec, NMR and traditional mass spec. It has demographics. It has, you know, clinical history, it has cognitive tests and so forth. And all of that has to be integrated together. So your question, Mehmet, is really how do we decide which of those factors becomes those that you use, for example, to diagnose Alzheimer's or predict conversion from mild cognitive impairment to Alzheimer's. And that process that we use involves no human expertise and no domain expertise. It is simply letting the data, the variables, compete for relevance with each other and interact in linear and nonlinear ways. So what you saw in terms of those math equations, most of the models are a little bit more complicated than that, but not much. We'll typically see, you know, maybe 5 to 8 variables used in a discrimination for a particular disease. Cancer algorithms tend to be a little more complicated because of subtypes and staging and so forth. But the variables themselves emerge from the process because those that aren't useful get deselected out of the evolutionary sort of population as it goes on. Does that make sense in terms of the question?
Yeah, it sort of, it was, in a sense, I expected it. It's sort of one of the things that we do is usually find the attributes through attribute importance or PCA analysis or something like that, which is not, doesn't really require domain information.
Yeah, we do none of that because those impose assumptions of linearity, variable independence, normal distributions. We don't want to do that.
Okay, I suppose you don't want to say how you're doing it, because that's probably under your IP.
I mean, I'm mindful of time, gentlemen and ladies. So what I'd like to do is ask one more question from the group, and then we're gonna move to breakout rooms. I mean, you know, one more question. Who's got a question? And otherwise, I mean, are there any drug discovery people amongst you that would like to ask those difficult questions?
If there's no one, I have one drug discovery question.
Oh, well, I think you might have won the prize. So you've got the prize. So one question from you, my friend.
Yeah, when Lucy was talking, I sort of wrote down a question. It says, where are we in automating drug discovery? I mean, we're talking about AI, and we're sort of talking about doing things that will help us. And there was someone who asked exactly how far did we come. I don't remember who that was. But how much have we actually managed to automate or partially automate with AI in drug discovery, especially?
I'm going to ask Jackie to answer that before we close this.
Yeah, but I mean, when you say automate, do you mean automate in terms of like the automated lab, or do you mean automate all the way through the process?
Oh, take this biomarker discovery, for example. You don't use anything wet, you use only digital stuff and only data.
I think there's loads of examples of where you are seeing a lot of automation. Like with our platform, it automatically triage, it looks at the data, but we still use the scientists to help then interrogate that data and triage it. So I would say there is a lot of automation all the way along, for example, not just in biomarkers, but also in the analytics of multiplex assays, in the in generative chemistry, in the iteration of structured design for multi-parameter optimization. So I think there are examples of automation. The whole process is not fully automated, and to some extent, I think that will be quite.
I think we should move on because I think this is a big conversation.
Yeah.
I mean, what I'm going to do now, I'm going to thank, firstly, Graham, I'm going to massive thanks to Patrick, Shahar, Peck, Jackie. Let's give them a big round of applause. I think there are more questions than answers. We'll be inviting you back, I hope, many times, because I think we need to untangle this because there's lots and lots of stuff here. And I think if I was the CEO of a company and wanting to invest in this, I'd be thinking, my goodness, what am I going to get out of it? So I think we're going to move into the next phase, which is the getting to know you. Graham?
I was just going to say, the way we're going to do this is we're going to do 2 breakout rooms of 15 minutes each. After the second one, we'll come back into the room, see if you want to do it one more time. If you want to do it one more time, we can do that. So we've got 45 minutes, but let's just see how it goes. So bear with me. I'll get to the breakout rooms, and here we go. Okay, so